Hello, and welcome to Back to the Science. I'm Dr. Susan Oliver, and I'm a scientist, and this is Cindy Oliver, and she's a dog. So last week, I made a video about a study that looked at a man from Germany who claimed to have received 237 vaccines, and 134 of them were actually verified. If you haven't seen the video yet, spoiler alert, there were no detrimental effects. Unsurprisingly, anti-vaxxers were not happy with this study because it is yet another piece of evidence that their scare stories about COVID vaccines are bollocks. And they let their displeasure about me covering it be known on the platform formerly known as Twitter. Here are a few examples. Get a refund on your PhD. Now, for anyone who doesn't know, you don't have to pay to get a PhD in Australia if you're an Australian citizen. Not only is it free, but if your marks are good enough, the government pays you a scholarship while you do it. And I qualified for the scholarship. Where are the pro-vax cultists at to defend Susan's mind-blowing analysis, happy, smiley, thingamy-bobbly? In desperation to be right and remain relevant, you've inadvertently proved our point. Thumbs up. If anyone knows what that means, please let me know in the comments. Cindy isn't impressed. You can see she's wandering off. What makes you think that people who don't use vaccines think about you at all? I don't spend my free time thinking about pro-vaxxers, but anti-vaxxers seem to be living rent-free in your head. Have you ever thought about getting a hobby? Says the person that I have never heard of who is replying to my post. Oh, you should worry more about being charged under Nuremberg Code than being told get your booster because people like you will see Nuremberg too. Yep, anti-vaxxers are still going on about that. Now, there were also lots of people pointing out that this is only one person and providing me with lots of anecdotes of people being fine after doing various dangerous things. Now, of course, if they'd actually watched the video, they'd know that I actually pointed out that the results were not generalisable and it was not recommended that anybody do this. But let's face it, most Twitter trolls don't watch my videos. They just leave nasty comments. Although I guess that's fair enough because I don't usually read their comments. I have my notifications on Twitter set so that I only get notifications from people who follow me. I do get to see other people replying to their comments, however, and I often like these replies, which then causes the original commenters to go nuts and complain that I'm ignoring them, which I am, because I have better things to do with my time than interact with trolls who don't watch my videos. But enough about that. This video is about a particular claim that was made by a number of anti-vaxxers. Here's an example from Amazeballs Shezza. Susan, the story is fake. Please keep up. You're an embarrassment to Australians. To which Councilled Mouse replied, don't take it personally. Susan is an embarrassment for mankind as a whole. Although I must admit, it must feel bad, laughing face, accompanied by a picture of a kangaroo because, as you know, kangaroos are everywhere in Australia. If I didn't have my door closed now, there would be a few of them sitting on the sofa with me instead of Cindy. Anyway, I was curious as to why people were claiming it was fake, so I decided to do a little more digging amongst the crazy comments, and I found this. Good God, you're naive. Jicky Leaks, Finderella One and Kevin McKernan and other Mouseketeers could soon dispel your illusion. But what's the point? So I decided to have a look at what these people had to say and found that two of them had already blocked me. So clearly they had no interest in educating me, but I hit pay dirt with Jicky Leaks. Susan, this story has been exposed as fake. We have confirmation from Kevin McKernan that the report has no validity. In other words, it's fake. Either that or they sequenced an unvaccinated person's T cells. And then there is a link to a substack which appears to be written by Kevin McKernan, which I mentioned on the last slide has blocked me. Anyway, I had a look at this substack and I decided it was worthy of a video because it is a perfect example of how anti-vaxxers misrepresent studies and leave out critical information to mislead their followers. So this is a substack. 217 jabs or legal jousting. Surgisphere level sophistry. Ooh, intriguing. Anyway, if you scroll down a bit, you get to the crux of their argument. The authors performed single-cell RNA sequencing on this person. Given Henner et al. Has found vaccine RNA/DNA in breast milk 5 days later, Castruta et al. Found vaccine RNA/DNA in plasma 28 days later, Crossan et al. Found vaccine RNA/DNA in heart tissue 30 days later. Gonzalez et al. Found vaccine RNA/DNA in placentas 10 days later. Surely there should be some vaccine RNA/DNA in this German individual, him, that took 217 jabs. And then they go on to supposedly show that there was no vaccine RNA in the individual. Now, if you're an anti-vaxxer, you probably find this argument pretty compelling. If instead you're someone who follows my channel, you're a bit more discerning. You know that just because someone claims something in a substack, it doesn't make it true. So let's have a look at each of the claims and see if the studies actually support them. Now, before we look at each study individually, I would just like to point out that although the substack keeps saying vaccine RNA/DNA, none of the studies mentioned found vaccine DNA in any person at any time point. And we have talked about this whole vaccine DNA thing before, so I will provide a link to the video if you want to have a look at it. Anyway, so the fact that the author, Kevin McKernan, included this RNA/DNA is just being deliberately misleading, but that's just the start of the deception. Okay, let's have a look at the first claim. Henner et al. Found vaccine RNA/DNA in breast milk 5 days later. So this is a study here, and what they did in this study was they took expressed breast milk samples from 11 women at various time points up to 5 days after they'd been vaccinated. And they used PCR to see if they could detect any vaccine mRNA in both the whole breast milk as well as various concentrated components of the breast milk. This is what they found. In 5 of the 11 participants, vaccine mRNA was detected in extracellular vesicles. And in 3 of these participants, it was also detected in the whole milk. And an extracellular vesicle is basically a membrane-bound nano-sized particle that carries things like protein or RNA in bodily fluids. They didn't find vaccine mRNA in any other components of breast milk that they analysed. They didn't check whether it was intact mRNA or just fragments, and they didn't find it in any samples taken more than 45 hours after vaccination. And for anyone who doesn't know, there are 24 hours in a day, which means the RNA was only found in the breast milk less than 2 days after the vaccine was given. So the claim that it was found 5 days later is total bollocks. On to the next claim. Castruta et al. Found vaccine RNA/DNA in plasma 28 days later. The title of this paper is SARS-CoV-2 Spike mRNA Vaccine Sequences Circulate in Blood up to 28 days after COVID-19 vaccination. So based on the title, this claim is at least true as far as the vaccine RNA claim goes. But of course, the title doesn't really tell the whole story. To start with, these weren't healthy people. They were patients with chronic hepatitis C infections. And vaccine mRNA was only found in 10 out of 108 samples. And there's more details in this pretty chart here. Each colour represents a different sample, and there are 10 samples in all where they found vaccine mRNA. But the bottom 2 samples come from the same patient at different times, which means that it was only found in, 9 out of the 108 patients. The x-axis shows which bit of the mRNA sequence they actually found, and the y-axis shows the number of reads they obtained for each sequence. And it's important to know that the y-axis is different for each sample. Also on the right-hand side of the chart is information on the time the sample was taken relative to their last dose of vaccine. Now, the first thing to note is that only one of the samples where they found vaccine mRNA was 28 days past vaccination. This is the red-coloured sample at the top of the chart. And as you can see, they didn't find a complete sequence of the mRNA. They just found a few fragments. In fact, they were only able to find the complete vaccine sequence in one of the samples they tested, and that is the magenta-coloured sample, which was taken 5 days after a second Moderna dose. So whilst a few fragments of mRNA were found in one chronic hepatitis C patient at 28 days post-vaccination, there is no evidence that this is common. Next, Krausen et al. Found vaccine RNA DNA in heart tissue 30 days later. So this is the paper here, Duration of SARS-CoV-2 mRNA Vaccine Persistence and Factors Associated with Cardiac Involvement in Recently Vaccinated Patients. And what they did in this study is they looked for vaccine mRNA in the tissues of people who had died after getting the vaccine. But just to be clear, they weren't vaccine-associated deaths. The vaccine was just a coincidence. So this is a heat map that shows where and when the vaccine mRNA was detected. If the box is white, it means no mRNA was detected. Grey means the results are unavailable, and blue or red means it was detected. And the colour shows how much. ALN stands for bilateral axillary lymph nodes, LV stands for cardiac left ventricle, RV stands for cardiac right ventricle, and MLN stands for mediastinal lymph nodes. As you can see, there were only 3 patients where it was found in cardiac tissue, and it wasn't found beyond 19 days, not 30 days as claimed. It was found up to 26 days in the bilateral axillary lymph nodes, but not in the mediastinal lymph nodes, liver or spleen. And it wasn't found anywhere beyond 30 days. As the vaccine mRNA was only found in the cardiac tissue of 3 patients, the authors looked further into this to see if they could find a cause. And what they found was that all the patients had healing myocardial injury at the time of vaccination and macrophage infiltration in the myocardium. So this is another claim that is total bollocks. mRNA was not found in cardiac tissue 30 days after vaccination and was not found at all in the hearts of anyone who didn't have a cardiac injury prior to vaccination. And finally, we have Gonzalez et al. Found vaccine RNA DNA in placentas 10 days later. Okay, this one's a doozy. Mr. McKernan must think his readers are gullible fools to have included it. You only have to read the title to know the claim is bollocks. The title is Minimal RNA Uptake and Inflammatory Response to COVID-19 mRNA Vaccine Exposure in Human Placental Explants. The key word here is explants. An explant refers to living tissue removed from an organism and placed in a medium for tissue culture. In other words, this is an in vitro study. They literally incubated the placental explants with vaccine in a petri dish. And even then, their results show that the human placenta does not readily absorb the COVID-19 mRNA vaccines, nor generate a significant inflammatory response after exposure. And as for the 10-day claim, that seems to have just been pulled out of McKernan's arse. It's just total bollocks. So now we come to the question posed. Surely there should be some vaccine RNA DNA in this German individual, him, that took 217 jabs. The answer to this question is a big fat no, because the evidence clearly shows that most samples taken from vaccinated people did not contain vaccine mRNA. So whilst it's possible that him would have some vaccine mRNA somewhere, it's definitely not a given. And in fact, when you look a bit further into it, you see that the chances are pretty unlikely. I'll explain why. This is how the substack explains what they did. Let's have a look at their scRNA sequence data. Since we put the Pfizer vaccine sequence in NCBI in June of 2023, it should be really easy to compare their RNA sequence data to the Pfizer vaccine sequence and see if any of it exists in this person. Stephen McLaughlin, along with another Anon researcher in this space, found the NCBI data for everything in their paper and aligned the reads to the Pfizer-BioNTech vaccine reference sequence we published in NCBI. The blue arrows on the bottom of the track are the spike ORFs. Yes, there are 2 overlapping ORFs, but that's a topic reviewed on a different substack. And by the way, when they say they found the NCBI data, they mean they followed the link provided in the paper. They didn't have to go searching for it. They go on. But there are 0 reads mapping to Pfizer-BioNTech spike. That's a big oof. Oof? You may recall earlier in the Substack, McKernan mentioned that the RNA reads came from single-cell RNA sequencing. In single-cell RNA sequencing, single cells are isolated and their RNA content is captured. And the cells that were sequenced in this case were T cells. This is the only data that McLaughlin and the other Anon researcher had access to. So let's have a look at the study that they mentioned that previously found vaccine mRNA in T cells. Oh, there wasn't one. In other words, they provided no evidence that vaccine mRNA was ever found in T cells. Oh, and one more thing. The T cells were isolated from a blood sample taken 189 days after him's last vaccination, which is more than 6 months later. A bit longer than the 30 days that vaccine mRNA has been found anywhere in human tissues. In the words of Kevin McKernan, that's a big oof. So, in summary, whilst the substack claims may seem valid if you take them at face value, if you look a bit more closely at them, you can see that they are total bollocks. There is no evidence whatsoever that the story is fake. Anti-vaxxers need to try harder. If you'd like to look further into the data I presented, I provided links in the video's description. And I'll also provide links to a few videos that look a bit more into the vaccine RNA and DNA claims that have been going around for a while. And please remember, this video is about the science, but you shouldn't take it as medical advice. For that, you should speak to your medical practitioner. If you've got this far, thank you for listening. And if you've liked, shared, or commented on the video, double thank you, because that helps the algorithm and means that more people will see the video. And of course, thank you to everyone who has bought me a coffee, or little Cindy here a treat. There we go, Cindy. There's a treat. There we go. She's having her treat. We really appreciate your support. We will be continuing to make videos about the science in the future. So if you'd like to join the cool kids and stay informed, please hit the subscribe button. Thank you.